Okay, so welcome. This is our first general meeting of the year. Under normal circumstances, I wouldn't talk in a microphone because I know my voice will carry, but we are recording this for posterity, so it's pretty cool. Today we're going to talk about ticks. We have a parasitologist that will be speaking a little bit later, Miss Molly Bechtel. Thank you, Molly, for joining us.
Okay. I am really excited to be here today to talk to you guys about some of my research and ticks and tick-borne pathogens of the Mojave Desert tortoise. And we don't have to worry about Borrelia burgdorferi here in the Southwest, but we do have ticks that carry a species of Borrelia that causes illness in humans. So we have these ornithodoros, these soft ticks right here. And they carry a Borrelia that causes tick-borne relapsing fever in people. And what happens with tick-borne relapsing fever is generally it's not an issue if you get it taken care of right away. But after about 7 days, after you've been bit by a tick, you'll start to have really high fever, headaches, body aches, symptoms that are very similar to a flu. And then those symptoms after 3 days go away and you feel fine. But then they come back and last for another 3 days, and that cycle continues unless you get treated with antibiotics. As I mentioned, generally it's you can take care of tick-borne relapsing fever very easily just with antibiotics. But if it goes untreated, it can cause more severe issues like neuropathy, and it does cause abortion in pregnant women. Regardless, they still occur here. We know that. They are nidicolous, which I'll talk about what that means in a second, and also opportunistic. So there are ticks that are specialists, meaning that they will just feed on one species of animal. But these guys, they don't care if you're a tortoise, if you're a bird, if you're a person. If you have blood, they will be more than happy to take a meal from you. And they're carriers of the causative agent of TBRF. It was discovered in the early 80s that 2 of the 3 species that occur in the Mojave, Ornithodoros parkeri and Ornithodoros turcata, are common ectoparasites of desert tortoises. So when they first hatch, they hatch into their larval stage. So the difference between larvae and nymphs and adults is that they only have 3 legs on each side of their body. They're also very small. So you can imagine they would be very difficult to identify at that stage, at least this species. Then they'll find a host, they'll take a meal, they'll fall off that host, and they'll molt into what's called a nymph. And oftentimes there's several nymphal molts, so they'll molt again and again, just into larger nymphs, until finally they'll get their last feed before they molt into an adult. And an adult is going to feed off of a host and then fall off that host and lay their eggs, usually in the same place where they were hatched. Okay. So this brings me to my research question, more or less. I keep mentioning that very little is known about these ticks and how they interact with these tortoises and possibly even public health. We don't know if the Borrelia that they carry is pathogenic to tortoises. We don't know if it's not. But we do know that they carry a harmful disease to people and that they like to lay their eggs and basically grow up in tortoise burrows. Blue belly lizards, which we have around here, but they're also in Northern California where Lyme disease exists. And they have been found to have what's called a borreliocidal factor in their blood. And what that means is that this enzyme, this protein in their blood, once it comes into contact with these Borrelia spirochetes, so in a tick gut, for instance, it kills it. It's just, so this is actually really cool because where you see more of these lizards in Northern California, a higher abundance of these lizards, you see less prevalence of Lyme disease. Because when a tick that's carrying Borrelia burgdorferi, the causative agents of Lyme disease, comes and bites one of these lizards, you can see this guy has a group of ticks he's carrying around with him. The enzyme in their blood will kill the pathogen. So then that tick will drop off of the lizard, go find a human to bite, and it's incapable of transmitting Lyme disease to that person because the blood in the blue belly killed the Borrelia burgdorferi. So it's really cool. And what's really interesting is that this enzyme is related to what's known as a thermolabile protein, which is also found in tortoises. But despite the fact that desert tortoises are commonly parasitized by ticks that carry a harmful pathogen to people, no research on the resistance or susceptibility in desert tortoises in relation to these ticks and the pathogen they carry has been done. So we don't even know if this could make them sick, but we also don't know if perhaps they're killing the pathogen that these ticks carry. So for future research, go ahead, go to the next slide. I'm going to continue identifying these ticks, but I think because they carry this pathogen that it's important it's done accurately. So what's done in medical entomology is a system called molecular barcoding, and it helps researchers identify the tick that a pathogen that this person is sick with. And basically that's just matching fragments of smaller DNAs to a larger genome and matching it up. So I'm going to modify this method and use this for identifying ticks because they're so difficult to key out. If you guys remember, go ahead and we're counting these tiny little bumps. And that's the only difference we have right now between the species. So comparing their DNA is a much more accurate way to do that, and it's used very successfully in medical entomology. Okay, the next one. I'm also going to refine my field collection method. So I do take ticks from people that collect them and find them on their tortoises. But I'm also interested in seeing if those ticks that are just hanging out in the burrows, not attached to tortoises, if there's any difference in the pathogen prevalence in those populations. So I mentioned the carbon dioxide traps, the dry ice traps, which don't really work that well. In theory, they do, but you have to constantly be out in the desert refilling the dry ice, so it's going into the burrow and getting the tick's attention. But what is done in some other entomology work is a leaf blower with the vacuum modification on it. And basically, you just add some tubing and you vacuum out a burrow, just like that. And then what's really cool about these ticks, so maybe you have like this bucket of dirt, you don't really have to sift through them because they will fluoresce under black light like scorpions. So that kind of makes it a lot easier to find them. So in order to determine if these tortoises do have that borreliocidal factor that kills Borrelia, I want to do an infection kinetic study, which is done non-invasively for the most part. The only thing invasive is a little blood sample from a tortoise. And if you haven't seen that done before, you actually go up under their carapace. It takes a few seconds, and it doesn't really seem to bother them that much other than the fact that you're pulling them out of their burrow. And then you take that blood sample and you put it in a petri dish. And in that same petri dish, you introduce Borrelia spirochetes. And you just measure survivability, which is exactly what that scientist did that determined that blue belly lizards have that same enzyme. So you just use their blood, pretty non-invasive, and I do think that tortoises probably do have this enzyme in their blood, and if they do, I think that would be really a win-win for desert tortoise conservation, as well as helping us learn how to manage a potentially dangerous disease across the landscape. And then finally, tick distribution and pathogen distribution has not been measured, has not been determined at all in the Mojave. So I'm going to take the ticks that people send me that I collect in the desert and model their distribution and their pathogen distribution as well. As I mentioned, it has been attempted for terracotta, but pathogen prevalence was never involved, and neither was the pathogen prevalence in Ornithodorus hermsii either when that was attempted to be modeled. So with that, I'd like to thank my collaborators, my lab, the folks at the USGS and at University of Nevada, Reno who have helped me collect ticks and analyze them. And with that, I'm happy to take any questions. Hey, great. Thank you guys so much for having me. I really enjoyed speaking today.
Thank you, Molly. Okay, everybody take a deep breath. All right, that was awesome information. Now, I want to let you guys know that this is something that came to our attention pretty much within the past couple of months. We have ticks at our habitat, and what we have done is we went crazy with it. We just tore up all of the burrows, put the pesticide down, and now we're in the process of putting the tortoises back. I don't want you guys to leave this room super worried. And the reason is that this is all brand new information, and I wanted to share this with you guys because this is what we're finding out as well. Right now I don't have a specific protocol, but you guys are asking the proper questions. What do you do when you see a tick? My advice right now is if you find a tick, give us a call. I think for academic purposes, we want to collect as many ticks as possible. How many have we given you so far, Molly?
15, 20?
Yeah, so I mean, obviously this helps with research, but if you get rid of your tick and you just take it off and kill it, that's cool. But give us a call, because if nothing else, we can share whatever information we have with you anyway, which we will do through emails and put it on our website and everything. But I don't want you guys to think that, oh my God, you can't stick your hand inside of your tortoise burrow, okay? Because this is, if you put it in perspective, I'm not going to say it's not important. It is important, but we just don't know. And I would caveat all of this by saying, I don't think any people have died from this in the Mojave. Yeah, there's black widows and all sorts of other stuff in burrows that you don't want to stick your hand in there anyway. So.
What I want to say also is that really the danger is that, as far as we know, doesn't really affect the tortoise. The tortoise will, it's annoying, but as far as we know, it's not harmful to the tortoise as far as just taking its blood. Just be more concerned about tick bite on you, and be aware that if you've got a tick bite and you have a fever, you probably need some antibiotics.
Yes, what Molly said was if you have a tick bite and you have a fever, go see your doctor. Go get some antibiotics. That's at the very negative case. But in the event that you do see a tick, give us a call. I mean, I'm working on a protocol right now for specifically this, and tearing up your burrow, I get it. If you find a tick, you want to take care of this problem. So give us a call, because if it does entail tearing up your burrow, well, we got volunteers to help with that, and that's, as far as I'm concerned, that's equally as important or even more important than building new burrows. So this is something that we are looking at, and we just want to keep you guys updated and have all the information out there. So it's really cool work what you're doing, Molly. It's very, very important work. Thank you for the presentation.